Today we're going to go ahead and start Chapter 1, which is titled Major Themes of Anatomy and Physiology. So there's a lot of terminology in anatomy and physiology. A lot of it are terms that are not commonly used in everyday life, partly because most technical terms in science are derived from Latin or the Greek languages. There aren't too many people anymore that use Latin. So a lot of the words are kind of foreign sounding. As a result, some biology professors have said in the past that taking an introductory biology class is very similar to taking a foreign language class because so many of the words are foreign. And so a lot of learning the language of anatomy and physiology is learning the meaning of foreign words. Okay? And we'll talk a little bit more about how words are constructed. If you understand how words are constructed in anatomy and physiology, it will make it easier for you to figure out what they mean without having to memorize a definition. If you can figure something out just looking at the root and the prefix and the suffix, then that makes it easier to learn anatomy and physiology. Okay. So, let me turn this on here. All right, so let's start out with some definitions. So, the word anatomy refers to the study of structure. And so for this course, since it's human anatomy and physiology, anatomy is referring to the study of the structure of the human body. And the word anatomy actually has a really interesting origin. The word anatomy comes from the word anatomize, which is a term that early anatomists used instead of dissection. So the word anatomy, which is the study of the structure of the human body, comes from the word anatomize, which means to cut or dissect, because that's the way that the early anatomists learned about the structure of the human body, was by dissecting or cutting human bodies so they could observe the internal structure. So anatomy literally means to cut or to dissect, and because that's the best way of learning human anatomy, is to study directly from the main subject of interest. So cadavers are often used for dissection as part of the graduate training and even at the undergraduate level of many institutions. Unfortunately, when our administration was planning this building, they made the intentional decision not to have a cadaver lab in this building. So we really don't have the facilities to do that. So unfortunately, we won't be able to do any study from cadavers. We will, however, do some dissection of organs. This semester, I usually get some sheep brains, and we dissect sheep brains and study their structure, because sheep brains, even though they're a lot smaller than a human brain, they have all of the same structures as a human brain. And if you can identify them on a sheep brain, you can probably identify them on a human brain. We also have several human brain models. So when you're dissecting and studying the anatomy of the sheep brain, you can compare it to a model of a human brain, so you can see what the differences are. Okay. That approach where you use organs from other animals for the study of human anatomy is called the comparative approach, and it's often used at the undergraduate level. Physiology, on the other hand, is the study of the functions of the body. And physiologists do more than just try to determine the purpose or overall function of an organ. They want to know more than that. They want to know how a structure or an organ functions. And that type of explanation of the function of a structure or organ is referred to as a mechanistic explanation, or the mechanistic approach, because most functions of the human body require the work of more than one structure or more than one organ that all work together in a system. And oftentimes the functions require many different steps. And so a mechanistic explanation is a step, it's a explanation of a step-by-step sequence by which the body functions to carry out one of the major functions of the body. And so some explanations in physiology are rather complicated because they involve many steps. The nice thing, though, is that you can break functions down into steps, and you can study the steps individually, and then you can piece them together to understand how the body functions as a whole. So even though they may be, and a lot of times the functions of the body also make sense because they do have an overall purpose. And if you understand the purpose, then you kind of know where that function is headed. You can predict what's going to happen. And if you can predict what's going to happen, you can kind of guess then how that's going to happen. So I had a professor one time in a physiology class that I took in graduate school that said that he liked physiology because he thought it was easy, because you can predict, you can guess what's going to happen. And most of the time you'd be correct. Now, there is more than one kind of explanation, though, in physiology and some of the other sciences. What I just described as a mechanistic explanation or mechanistic approach. That describes a step-by-step sequence of events by which a process occurs. So most of the mechanistic explanations answer questions that begin with the word how. It explains how a system of the body functions. There is another type of explanation, though, in physiology, and that's called a teleological explanation. Teleological explanation answers the question of why a structure or a process exists in terms of its purpose or its reason for existence. Okay, so one example of a teleological explanation would be an explanation of why we have lungs, or what are the overall functions of the lungs. And so a teleological explanation would say that the function of the lungs is to provide the body with oxygen. And that would be correct. That's the main function of the lungs, is allow oxygen to be absorbed into our body. However, physiologists want to go beyond that. They want to know more than just the purpose of an organ. They want to know how it works. Okay, so a complete mechanistic explanation for how the lungs work would be fairly complicated. You would have to describe all of the muscles that attach to your rib cage and your diaphragm, which contracts when you breathe. You'd have to describe the volume changes that occur in your thoracic cavity when those respiratory muscles contract. You'd have to describe the pressure changes and how that affects the volume of the lungs. And when the volume of the lungs change, that changes the pressure in the lungs, and that causes air to flow and enter the lungs. So we're going to learn about that eventually when we get to the respiratory system. I'm not going to go through all the details of that mechanistic explanation right now because it's a little premature. That's just an example of the complexity of some mechanistic explanations. However, having said that, this first semester of this two-semester sequence is going to focus a lot on just the structure of the human body, because this semester we're going to go through the skeletal system and all the muscles. There are a lot of bones and there's a lot of muscles in the human body. There's typically 206 bones, although the number varies from individual to individual. Most people actually have some extra bones. If you look at the skeleton of a newborn or even of a young child, they have a lot more bones than an adult does, because some of the bones fuse during the growth of a child. So there's a lot of structure to know in the musculoskeletal system. So we'll be focusing a little bit more heavily on the physiology in the second semester than we do this first semester. We are going to go through things like how muscles contract this semester, though. So we will get into some physiology, but a lot of, especially what we do in lab, is going to be more focused on anatomy this semester. We will do some experiments in physiology in lab, but it's just the way it works out. It's not an intentional decision. It's just that the organ systems we cover are more complicated structurally in the first semester than they are in the second semester. So we can spend a lot more time on the physiology, which really to me is the more interesting part in the second semester of the class. All right, now the first major theme of anatomy and physiology that I think is important to understand is the complementarity between structure and function. It's one of the reasons why we teach an integrated anatomy and physiology course. Complementary of structure and function means that you can't really explain the structure of the human body or the functions of the body in isolation, because structure and function of the human body go hand in hand. Okay? So even back when I used to teach just separate human anatomy and human physiology courses, I still talked about function when we're talking about anatomy, because if you don't, you can't really understand what shapes the structure of the body. If you don't talk, it doesn't make sense. Anatomy without the function doesn't make sense. So in a way, it's a lot easier to talk about both the structure and the function at the same time, instead of doing that in different semesters. Functions of our body also, over long time periods, shape our structure through natural selection or through artificial selection. So if a person inherits a trait that doesn't work quite as well, because there is a lot of variation from individual in our traits, then that may make them less likely to survive to a reproductive age. I mean, they may die early. And if that happens, then they're not going to pass those genes on to their offspring. Other people that have more favorable variations are. And so gradually, over millions and millions of years, our body becomes adapted to functioning like a well-tuned machine. Okay? So structure and function go both hand in hand. Structure determines the function. Structures are what carries out the functions of the body. Structure's kind of like the parts of an engine. It's what causes the engine to work. But if the parts don't work together, you're going to throw that engine away. You're not going to keep that. And so gradually, the engines that we have left are the ones that do work and usually work very well. So anatomy and physiology can't really be completely separated. I think it makes a lot more sense to use this integrated approach. Now, there are a lot of different study methods within the field of anatomy and physiology. And these are different approaches that anatomists and physiologists use to study the human body. Because keep in mind that science is a process. It's not just a set of facts. And so even anatomists go through a process of learning new things about the structure of the human body. And our views about the structure of the human body change. Just a few years ago, there was a couple of anatomists that did a study of the quadriceps muscles, quadriceps group, which is in the anterior part of your thigh. And they dissected both legs on something like 7 or 8 different cadavers. And they were looking to see if part of the vastus intermedius, which is a deeper muscle in the thigh, actually was a separate muscle, because it was noticed by some anatomists that part of that muscle looked like it has a completely separate tendon that attaches to the knee. And so they dissected these 7 or 8 cadavers, both of their legs, and they found in every case there was actually a completely separate part of what they thought was the vastus intermedius muscle, but it had its own separate tendon that attached to the patellar tendon that attaches to your patella. And so they came to the conclusion that there's not 4 muscles in the quadriceps group. There's actually a small 5th muscle. So we probably shouldn't even call that the quadriceps group anymore. But you probably won't see that in your textbook, because sometimes it takes a few years to get new information like that in textbooks. It's a real small muscle though, and it's called the tensor vastus intermedius, I believe is the name of it. But they probably don't even know what it does. It probably just helps to keep the vastus intermedius in line. Probably just kind of pulls it to one side and make sure that it's lined up correctly. So that's just an example of how science changes, how our views of things change. So one method or technique that anatomists use is called gross anatomy. Gross anatomy is one of the oldest approaches to the study of human anatomy. Gross anatomy refers to the structures visible to the naked eye. Okay, so gross actually comes from the German language, which it means large. So they're structures that are large enough that you can see without the use of a microscope. And there are several different approaches or techniques that are used in gross anatomy. One of them that I already mentioned is dissection. Dissection is a careful cutting and separation of tissues to reveal the deeper structure of the body. And a lot of times when you're doing a dissection, you don't even have to use any sharp instruments. Sometimes it's just like separating the muscles, and you can sometimes just kind of tear through the fascia with a blunt probe. Part of the problem with using a scalpel or scissors is it's easy to accidentally cut things that you don't want to cut. So anatomists use what's called blunt dissection sometimes, using a blunt probe to kind of tear through and separate tissues. Another approach is inspection. Inspection can be used to study either the internal anatomy or the external anatomy. Inspection means just a look. And you can see a lot of things on the surface of the body that's part of our structure. And you can even determine a lot about a person's health just by looking at their external appearance. If a person's liver is not functioning properly, a person may have yellow jaundice. Their skin and the whites of their eyes might have a yellowish color. Or if they have an infection, there may be a kind of a red area where the infection occurs. So you can determine things like that just by inspection, just by looking. You can even use inspection to study the internal structures, but you have to do that in conjunction with other techniques, either radiographic techniques or dissection. Another method of studying gross anatomy is palpation. Palpation means to feel. So one good example of the use of palpation is determining a person's radial pulse. By putting slight pressure over the radial artery near your wrist, you can determine a person's pulse, and you can determine their heart rate from that. That's an example of palpation. Okay. Doctors will sometimes, palpate like around a person's neck to feel if they have swollen lymph nodes, because sometimes a swollen lymph node or swollen salivary gland is a sign of disease. Another approach to gross anatomy is percussion. Again, doctors sometimes use percussion to determine things like if a person has pneumonia. Percussion refers to tapping on the body and then listening to the sounds that are produced. So if a person has pneumonia, fluid might accumulate in their lungs. And so if you tap on a person's chest, if there's fluid in the lungs, that's going to produce a different sound than if there isn't a lot of fluid in there. If your lungs are normal, when you tap on a person's chest, if you listen to the sounds with the stethoscope, it's going to produce kind of a hollow sound. But if a person has a lot of fluid in their lungs, it's not going to sound the same. So percussion refers to listening, or percussion refers to tapping and then listening to the sound. Another method is auscultation. Auscultation just refers to listening of the sounds that are produced when the body functions. The most common example of that would be listening to heart sounds using a stethoscope. Okay, you can determine sometimes if a person has a leaky valve in the heart by listening, because you can hear the blood leaking through the valve when the heart contracts. And then I already mentioned this earlier, comparative anatomy refers to the study of other animals in order to learn about the structure of the human body. Comparative anatomy goes back a long ways, well over 1,000 years, probably more than 2,000 years, because during the early years of the study of human anatomy, there were laws against dissecting cadavers, because the church considered that desecration of the body, and so anatomists weren't allowed to, at least they couldn't do it out in the open. They weren't allowed to dissect cadavers. So they learned about the human body through the dissection of other animals, like they might dissect a dog or a cat. And this technique is still used today in undergraduate laboratories. When I took comparative anatomy as an undergraduate, we dissected a cat, for example. The problem with the comparative approach is the cat is not a human. There's some things that are different between a cat and a human. And so some of the early anatomists made mistakes because they assumed that some of the internal structure of a cat was the same as it was in a human. And so, it took anatomists sometimes almost 1,000 years before those mistakes got corrected. Nonetheless, it still can be a useful technique because there are some organs of the body, like the heart, that are very similar in all mammals. Heart of all mammals has 4 chambers, for example, and there's 4 valves in the heart. And so you can learn a lot about the human body by, say, so for example, we're going to use the comparative approach to a limited extent in this class. In the second semester, I usually get some pig hearts to study the heart, because pig hearts are about the same size, same shape, and have all the same structures as a human heart. Because pigs are mammals, and we're also mammals. All mammals have a similar 4-chambered heart. So you can learn a lot about some parts of the body at least, using the comparative approach. This semester, we're going to do a dissection of a sheep brain. And the sheep brain has all the same structures as the human brain. It's just quite a bit smaller, but it still has the same major regions, the same overall structure. Another approach to human anatomy is radiological anatomy. Radiological anatomy uses imaging techniques like X-rays or CAT scans or MRI or PET scans or sonography. X-rays use electron beams which can penetrate soft tissue very easily, but it's absorbed by hard tissue like bone. And that produces a radiographic or X-ray image that you can use to examine for defects of the skeleton. It doesn't work very well though for looking at problems with soft tissue. So other techniques like MRI is used. MRI is machines are a lot more expensive, and so it's a lot more expensive technique. But you can observe problems in soft tissue. PET scans use radiation. The way a PET scan works is they inject a radioactive solution into your bloodstream, and they often use that to check to see if a person has cancer, like a small tumor in some part of the body. And the way it works is that if a person has a small tumor, tumors take up nutrients, and they usually have a larger blood supply than normal tissue. So when you inject that radioactive solution into a person's bloodstream, cancer cells will take up that radiation more quickly because they usually use something like radioactive phosphorus isotope. And phosphorus is an essential element for cells. And so cancer cells will take up that radioactive solution more quickly than normal cells. And so then they can detect if there's a tumor in the body. If they see a concentration of that radioactive element on an X-ray film, they know that there might be a tumor in that location. Sonography is the use of sound waves, like ultrasound. They use that to examine human embryos while they're still in the uterus, or human fetuses. They could also use it for other purposes, for soft tissue problems. Then finally, the last approach to the study of human anatomy and physiology, which is one of the more newer approaches, is microscopic anatomy. And microscopic anatomy didn't come into use until about the middle of the 16th century, about the middle of the 1700s. Because microscopic anatomy requires the use of a microscope to see structures of the human body that are too small to be seen with the naked eye. So there's kind of two different levels of organization that are studied using microscopic anatomy. The first is cytology, which is the study of cells. Cells are the smallest possible functional unit of the body that can carry out life processes like reproduction and metabolism and cell division. And all cells are usually very, very, very small, so small you can't see them with the naked eye. So until the microscope was invented, biologists couldn't study cells. They didn't even know they existed until the microscope had been invented. The other level of organization above the level of the cell is the tissue level. The study of tissue is called histology. And tissues are kind of the building blocks that are used to produce organs, which are larger structures that are visible by the naked eye. So tissues are defined as groups of cells that are similar in structure and function. They're groups of similar cells that work together to carry out a similar or the same function. And there's 4 major tissue types that are classified by biologists. There are tissues that form sheets of cells. They're called epithelia. There are connective tissues that help to support and protect and connect different parts of the body together. There's muscle tissue, which can contract, which can allow for movement of various body parts. And then there's nervous tissue. Nervous tissue is a tissue type that can produce neural impulses that can allow communication of different parts of the body. And then within each one of those 4 major tissue types, there are different subtypes that have the same general function, but they may vary slightly in their location and exactly what they do. And so altogether, there's a little over 20 different specific tissue types. That will be the subject of our laboratory that we do next week. So this week, we're just looking at some of the general organization of the body, the cavities and the organs and organ systems, and some of the terminology. Next week, we're going to start looking at we're going to do all the tissues in 1 week. So that one is probably a little more difficult than the first lab that we do, because not every student in this class has probably looked at tissues before, because it's not something you can see with the naked eye. So unless you've taken an anatomy and physiology class before, you probably haven't studied tissues very much. You might have been introduced to them a little bit in a freshman level biology course, but you just don't spend a whole lot of time on it. So we'll be going through the tissues next week. So, and we'll have to use microscopes to do that, because you can't see tissues without a microscope. All right. So, next part of this presentation, I had some of those clap questions, but thus far it doesn't look like it's going to work today, so I'll have to get that figured out. So we'll have to. I'll come back to this maybe at the beginning of class on Friday once I get it worked out. Okay. So, the next part of this chapter, we're not going to have time to go through this whole section of chapter 1, but I can at least give you a little introduction to it. The next section is about the scientific method. And I think this is important because you need to kind of keep in your mind that anatomy and physiology isn't just about learning facts. It's a way of knowing, it's a way of understanding the structure of the body and how the body functions. And the scientific method involves facts, laws, theories, and there's also a 4th term, there's hypotheses. Okay? So we'll talk about how scientists go about learning new things about the human body. Okay? Now, in the course of using the scientific method, there are 2 different types of what they refer to sometimes as higher level thinking or critical thinking. These 2 forms of critical thinking are referred to as inductive and deductive reasoning. And in the course of using the scientific method, you have to use both inductive as well as deductive reasoning. Okay? So, inductive reasoning is a process of taking many different specific observations and then putting those separate observations together to make a generalization about maybe how something works or how something is structured. Okay, so let me give you an example. Suppose you're an anatomist and you do a dissection of 10 different cadaver arms, and you observe a lot of variation in the structure because no two people are exactly the same. Okay, so you're making specific observations on 10 different arms, and then you want to describe the typical structure of that organ. Well, after you make those 10 observations, you think about what do all those structures have in common, and then you describe what that has in common. That's an example of inductive reasoning, because you're making a generalization from specific observations. Okay? And this is how a lot of scientific theories are constructed. Scientific theories are usually constructed by different scientists carrying out different experiments that are related. And then from that, from the conclusions of those experiments, a generalization is made that describes how something functions. And that involves the use of inductive reasoning. Okay, so theories are made through inductive reasoning. Another example is that early on in the history of science, different scientists made observations in different kinds of organisms that they were composed of cells. Robert Hooke was the first scientist to observe cells in plants. And then Schleiden and Schwann observed that animals contain cells. And so together they put those specific observations together to form the generalization that all organisms are composed of cells. And we now refer to that as the cell theory. Okay, so that's another example of inductive reasoning. Deductive reasoning is kind of the opposite. Deductive reasoning takes a theory, which is a generalization based on experimentation, and then making a prediction in a new situation. Okay? So, an example of deductive reasoning, let's say you discovered a new kind of germ or pathogen. Pathogen is a disease-causing germ. And you don't know what it is, but you can make the prediction, well, if that organism is made up of a cell, at least one cell, then that organism has to be a living organism, because all organisms are composed of cells. And so you can take a look at that pathogen with a light microscope, or you can take a look at it with an electron microscope, and if you see it has all of the major parts of the cell, then you can conclude that it's living. Okay. So deductive reasoning takes a theory or generalization that's supported with experimentation, and then making predictions from that in a new situation. Okay. So deductive reasoning is what scientists do when they carry out experiments. Because when you do an experiment, you're testing a hypothesis, which is a tentative explanation. And you make a prediction, and then you carry out the experiment and you see if that hypothesis is supported by the results or not. And if it is supported, then you accept that hypothesis and you take that as the best explanation. If your results don't support your hypothesis, then you reject it and you throw it out, and then you come up with a new hypothesis. Okay. So Deductive reasoning is what you do when you're testing hypotheses, which is basically the way you carry out the scientific method. So, scientific method is sometimes called the hypothetico-deductive method because you start out with a hypothesis, which is a tentative explanation that may or may not be correct, and then you test it using deductive reasoning by way of experimentation. And so a hypothesis, and the plural of hypothesis, hypotheses, is a prediction that may or may not be true. Okay. So what scientists do then is they carry out an experiment to test that hypothesis. And one very common method of carrying out an experiment is to have two or more groups of experimental subjects, which are the organisms you're studying. One group, you keep all the variables constant. And then in one or more other groups, you keep all of the variables the same as in the first group, which is called the control group, except you change just one variable. And then you observe the effect of that change. And in order to determine if that change in one variable does have an effect on your experimental group, you have to have a control group where you don't change any variables. Because without the control group, you have nothing to compare your experimental group with. So if you see a change in your experimental group, if you don't have a control group to compare it to, you don't know if that change that you observe is due to that manipulation or if it was just a matter of chance that you saw a change because the animals maybe got older. Okay. So that's not the only way you can do experiments, but that's a very common experimental method. All right, well, we're kind of running out of time, so why don't we stop here? On Friday then, we'll talk about some of the features of valid scientific experiments, like sample size and controls, and then I'll have you work on developing an experimental design for a situation. So,